Hello everyone. So in this video, we are going to cover the components of the adaptive immune system in details. We have seen in the last video that when the innate immunity fails, the adaptive comes into play. The lymphocytes, namely B and T cells, along with antibodies, takes in the charge. But how does it identify the foreign invader and elicit the appropriate response? The first important aspect is that it has to identify the antigen. So before going further, we should know what are antigens. As the name specifies, these are anti plus gens, means the substances which produce antibodies or leads to the generation of antibodies. Antigens can be proteins, they can be polysaccharides, they can be lipids. It can be a pathogen or any large molecule that does not belong to the body. So let's first understand the components of adaptive immunity and how do they tackle the antigens or pathogens. The body identifies the antigens and captures and presents to lymphocytes for the appropriate action, and this is known as presentation of antigens, which is being done by antigen presenting cells. And second, the action which is being mediated by the lymphocytes, namely B and T cells. So when we're going to talk about the components of adaptive immunity, we just classify them into three components: MHC complex, which are involved in the antigen presentation, and the rest two components include the special responses either in the form of cellular response or the humoral response. So let's first move to the first component of adaptive immunity, that is the MHC complex, which includes the important cells called antigen presentation cells or APC. Now, all the nucleated cells usually present self-peptides which are derived from self-proteins, and these are the MHC ones. So that means they have their names which can be identified and they are marked as self cells. So MHC complex basically plays a major role in directing the adaptive immune system. They are actually group of genes that encode glycoproteins which are present on the cell surface. Okay, just like this. The surface of all cells in a combination of almost unique for each person. And these cell surface glycoproteins are very necessary for cell self-recognition and prevention of immune system targeting its own cells. Now, on the basis of their properties, they have been classified into two types of MHC molecule, the class 1 MHC and class 2 MHC. So when we talk about the class 1 MHC molecules, these are the glycoproteins which are present on all nucleated cells on the body. Rather, they are not present on the red blood cells or platelets. These class 1 MHC molecules are made up of alpha chains with 3 domains. So here you can see this is alpha 1, this is domain 2 alpha 2, and this is third domain alpha 3, which are attached to beta 2 microglobulin unit. When they combine together, they form an antigen binding cleft. MHC class 1 plays essential role in recognizing specially virus infected cells, cancerous cells, tissue graft during transplantation. The reason being, as the cell machinery is altered during these phenomena, whether it is virus infected cells. So when virus gains an entry inside the cells, it altogether hijacks the cell's machinery. Cancerous cells altogether alters the working of a typical cell, and tissue graft during the transplantation also involves the replacement of a donor's tissue within the body. So basically these MHC1, apart from identifying the cell also help in recognizing the virus infected cells, cancerous cells and tissue grafts. The second type is MHC2 proteins. Now these MHC2 glycoproteins are present only on macrophages, dendritic cells and B cells. All three of these cell types are capable of phagocytosis, and their function is to engulf the antigen that originate from outside the cells. So class 2 MHC molecules is basically made up of an alpha chain and a beta chain with only 2 domains, alpha 1 and alpha 2, beta 1 and beta 2 respectively. Here you can see. And then after the combination of two chains, they form an antigen binding cleft where the antigen is being presented. So let's see how these antigens are processed in the class 2 MHC molecule. So after the pathogen is being phagocytosed, it is broken down and results in the peptide fragments in the phagosome. Now these peptide fragments goes and binds with the MHC2 molecules and finally presented on the cell surface. These cells act as antigen presenting cells and present the antigen to other lymphocytes. So here the two categories of MHC molecules where they are made up of different domains forming two different categories which identifies and recognize different cell types. Moving to the next two components of adaptive immunity, which is mediated by lymphocytes as cellular response and humoral response. The lymphocytes are free-floating T and B cells in the lymphatic system and spend most of their time in the lymph nodes. These lymphocytes must recognize antigens presented by the antigen-presenting cells and also learn self-tolerance, meaning they must not attack the body itself. Let's begin our understanding with the cellular response first. So cellular response is mediated by T cells where they acquire the ability to kill the pathogens. These T cells originate as hematopoietic stem cells from the red bone marrow. The population of these hematopoietic stem cells then infiltrate into the thymus, where they undergo maturations to become the functional T cells. Here in the thymus, they are dividing further to mature into active T cells, and this process is known as T cell maturation, which is controlled by the hormone thymosin. The cells differentiate into diverse and complex type and form into three major types, which is CD4 T cells, CD8 T cells and CD25 T cells based on the surface markers they possess. Before moving further, let's understand what is meant by this CD or what does the CD means in terms of T cells. So the T cells or the T lymphocytes cannot be distinguished morphologically since they are similar in appearance. However, these lymphocytes express different types of cell surface molecules which have specific functional roles. These cell surface markers are protein molecules that provide information about the cell type and they identify and discriminate between different cell populations. These CD molecules act as both signaling adhesion molecules which have biological and diagnostic significance. Now, how does it provide information about the cells? These molecules are identified by different antibodies specific for different epitope. This group or cluster of antibodies that bind to each protein molecules is termed as cluster of antibodies, and therefore these specific cell surface markers are known as cluster of differentiation, which is abbreviated as CD. So let's now understand how these molecules are designated. So there is a prefix CD which is followed by a number. The number reflects the order in which the cell surface marker were discovered at present, and CD markers range from therefore CD1 to CD371. This CD nomenclature is useful in identifying many different types of leukocytes. The CD designation tells about the cell lineage species to which the cell belongs and stage of maturation or activation of the cell. Now let's move back to the maturation of T cells. Now a proportion of these mature T cells continuously migrate from thymus into the blood and other lymphoid organs, maybe lymph node or spleen, where they play a major role in the body's specific immune response. The antigen-presenting cells in the lymphoid organ present the antigens to these mature T cells where they proliferate into different subset population. Now let's see what happens inside the lymph node. So this is here how the antigen-presenting cells binds with the typical T cells via their T cell receptors and leads to their activation. And whenever they encounter a specific antigen, they proliferate into their different effector T cells. So upon activation, the CD8 cells proliferate and differentiate into cytotoxic T cells. These cytotoxic T cells are responsible for killing virally infected cells. On the other hand, CD4 T cells proliferate and become T helper cells. Upon activation, they release cytokines and activate B cells, T cells, and macrophages. The T helper cells also present antigens to CD8 T cells. So that's all about for the cellular response. In the next video, we will going to see the next component of adaptive immunity, that is the humoral response.